thank you very much for inviting me to speak at Rose. Thank you, Kim Marie. I'm really honored and delighted to be here. It's terrific. I've learned so much already. So what I'd like to do today is tell you a little bit about the biology of the immune response in breast milk. And my goal is that you'll understand this response and be able to, at the second part of my talk, to be able to participate in helping us develop the best study possible for African American women. So to make this really meaningful for all of us, I think if you have any question or any comment, you should interrupt me at any time during my talk. Okay? Great. So the immune response in breast milk has three major components: antibodies, cytokines, and immune cells. And I know that a lot of you, all the lactation consultants, might know a lot more about this than many of us. But I want to go back and just review the basics, just bring us all to the same page. So the antibodies, also called immunoglobulins, produced by those immune cells. What's important, I'm going to go through the three classes of antibodies, and it's actually very important because this is one way that we all differ, that people can differ. And so IgA is about, or immunoglobulin A, about 90% of the antibodies in breast milk are immunoglobulin A, and it becomes important. About 2% of the antibodies are immunoglobulin G. It's just a class or a type or a flavor. And then the third one is immunoglobulin M, and about 8% of the antibodies in milk are this. So in response to SARS-CoV-2, we're going to produce specific antibodies in our milk that are slightly different than those in our blood, right? In our blood, 75% of our antibodies are going to be this type IgG. Okay, let's go on and take a look. I know you guys all know this, you've seen it many times. This is the SARS-CoV-2 virus. And what you see here, these red proteins, heart-like shaped proteins, that's the spike protein. It's also, if you look at it, it's what gives all coronaviruses their name. It's a crown, right? You see the crown going around, so it's a coronavirus. What's important here is this, I've highlighted it, the spike protein. So for all of the researchers who study the immune response, we're measuring an antibody that's specific to that spike protein. And so are all of the people who measure a response to the vaccine. They're measuring an antibody that binds specifically to that protein. And that's because the vaccines were made against this spike protein. And just a little bit more about this, because if you look in any of the literature, you're going to see this. There's a particular place on this little crown-like protein of the spike protein called the receptor binding domain. And that's exactly the bit of protein that antibodies are made against, all the ones that we measure. So remember I said there were three classes of proteins. So if you think of those three classes, then we're going to have anti-SARS-CoV-2 RBD IgA, and that just means it's against the SARS-CoV-2. It's specific to that receptor binding domain, and it's called IgA. And you might, if you get a test, you might see this result in a test. And then we have, of course, the anti-SARS-CoV-2 RBD, receptor binding domain IgG, and the third one, the anti-SARS-CoV-2 receptor binding domain IgM. All of the antibodies that are measured against milk or in saliva or in baby's tears or in blood are against these three antibodies. Okay, in both infection and vaccination. This becomes important at the very end of my talk. Okay, so if I measure the levels of antibodies in your milk, does that mean that those antibodies can destroy the virus and neutralize it? And it's maybe yes and maybe no, but there's a second assay that sometimes it's done, and if you look in the literature, you'll see it in some cases, and it's called a neutralization assay. And here what you want to see, this is the typical shape of an antibody. It always has that shape. You see it's binding to that spike protein, exactly on that RBD, but it's binding to the spike protein. And the real question is, does this antibody here, either IgA or IgG, because IgM doesn't do it too well, does it prevent this virus from attaching here and getting into your cell and making you sick? And so that's another way of saying the antibodies can't just be present, they have to work. So in some studies, we not only want to measure the antibody level, but the neutralization ability. And we do it with two assays. They're either called cell-based assays or inhibition assays. Okay, so we finished the antibodies, right? Just to summarize, I will tell you that higher levels of anti-SARS-CoV-2 IgA and IgG in milk are considered protective, and that we have neutralization assays that show that the antibodies are working, and they're both inhibition and cell-based. Interestingly, no one has tested in milk to see whether these two assays give the same response. Okay, maybe you'll remember that, okay. Okay, the second component are the cytokines, right? We did the antibodies, now the cytokines. The cytokines to the immune system are equivalent to the hormones in the reproductive system. They communicate back and forth. So we're particularly interested in T lymphocytes and the cytokines that they both secrete and that contact them. And this is how the immune cells talk to one another, and they also talk to your other cells in your body. And so you might ask, like, okay, so I forgot to mention, they have names, and you're going to see some of these funky names like interferon gamma, interleukin 6, interleukin 8, and tumor necrosis factor alpha. So those are the names of just some of the cytokines. And you have all heard about cytokine storms and how that makes us so sick, right? We get this big influx of cytokines. They cause a big inflammatory response. But why do we care about it in breast milk? We do care because the cytokines in milk are important for the development of the infant immune system and also the infant digestive system. So extra cytokines are sometimes actually quite good, and they also can have a negative effect. So we want to monitor the cytokines in milk. All right, and a third component is the immune cells. So we have cells in milk. It's very controversial what these immune cells in milk, they're called T lymphocytes, but they have memory, and sometimes long-lasting memory. These are the cells that can remember that they were exposed to a virus 6 months ago or 9 months ago. And so we can measure these immune cells also in breast milk, whether or not they stay in the infant's gut, whether or not they ever get out from the infant's gut into the circulatory system, whether they get systemic. That's a very controversial field. But they are present, we can measure them, and there's some thought that they're doing some benefit for the infant. Okay, so we've just finished the background. I actually have 3 parts to mine: background, then my research, and then we'll talk about the study. So any questions right now before we look at some data? We're going to use those 3 factors of the immune system, the cytokines, the antibodies, and the cells, to go look at what people have done. No questions? All right, let's look at some data. Antibodies, cytokines, and immune cells. So, as was mentioned, I'm really a breast cancer researcher, and I study breast milk in hopes of finding ways to prevent breast cancer. But with COVID-19, we figured we better do something and women were actually asking us, aren't you going to look to see what's in our milk? Because initially there was a big fear that the virus was in the milk. And I think you all know the virus is almost never in the milk. And you should breastfeed, and a woman can breastfeed because it's extremely unlikely that she would give COVID to her baby through her breast milk. It's much more likely that the kisses and the breathing and everything else would give COVID to the infant. Okay, so we were fortunate to collaborate and do a study with our colleague in the hospital, and we collected colostrum from 15 women who were either identified by a PCR test, so they know that they had SARS-CoV-2 when they came into the hospital, and 2 of the women actually had a PCR test months previously. And so the women had COVID, 15 women. Colostrum is so, as you know, so valuable, we didn't really want to ask the women to give much. So we asked them to put a few dots of their milk, to put these paper cards up to their breast and to put milk on that. And then 4 women also, we actually asked all women, but 4 on the second day had enough milk that they put some milk in these little containers. The reason why we had to do that is we didn't really know whether we could measure antibodies and cytokines from this piece of paper. So we set up the study, and as you can see here, we did a dilution of the whole milk to sort of equalize what we thought we were eluting from this piece of paper in a saline buffer. And what you see over here is that for each of the 3 antibody classes that I mentioned, whether we're looking at the spot card eluate here, or the liquid, you see a very similar response. Some difference. This woman has higher in her spot card, so whatever we did, maybe her milk was a little thicker on the paper and we didn't get that dilution correct. But, and here she is there. But in general, this was the first study that said, you know, you can use a piece of paper. You can put a piece of paper, it's a special paper, but not up to a woman's breast, and measure cytokines and antibodies in the milk from her breast. And so this has the potential to do really large-scale studies, if one wanted, of thousands of women. Okay. Now, this truly is a very busy, complicated slide, but I just want you to focus on a few things. So first, I want you to see that in this row here, it's the participant number, so there should be 15 of them going across. And in this row up here, L and R means for their left and right breast. And we think it is really important always to take milk from the left and the right breast and keep them separate in analyses, because that will help us understand if there are different things occurring in the left and right breast. Now, the next thing to note is this dashed line. And so that anything below that dashed line is not a significant increase in antibodies, and anything above it is a significant increase. So if I had to tell you the major take-home of this slide, it's that women vary. Women really do vary. So you can look at their IgA, their IgG, and their IgM, and they vary. So here, if we look at participant 15, so here, she was first tested positive 143 days prior to delivery, and her symptoms were 144 days. The last time that she was positive was 116 days prior, and she has a very robust, long-living antibody response. In comparison, participant 14 was still tested positive with a PCR test at delivery, and her first positive test was 98 days previously, so it might have been a second infection, but she has very low response. So when you think of when people get sick, some people get very sick with COVID, some people don't get very sick with COVID, some people think they've never had COVID, and then they do an antibody test and they have antibodies. You can see that here, that all these women giving us colostrum, we gathered their data, they have very different responses. Any questions? No? Okay. So let's go back to the take-home message for this study here, is that, again, this is the background line. So the answer is that colostrum from most women had anti-RBD antibodies, and IgA is the biggest response. And that makes sense because I told you in milk, 90% of the antibodies are IgA. Now, of course, we did want to test our assay, and so we went back, and we're fortunate that these colostrum samples came from our previous studies. They were actually done in 2013. And there are only 8 women there, but as you see, there are some quite above the line. So it was 2 women who had both their left and their right breasts were above for both IgA and IgG. And so you might say, well, what's going on? Well, keep in mind that it's a coronavirus. We've had other viruses, and it's called cross-reactivity. So we don't really know, but it's possible that some of the reasons why many people have a lesser response is because they've had other infections in their past that make them now have these antibodies still present. Okay, so they have some antibodies, they have cross-reactivity. Maybe it's not as robust, but they have some cross-reactivity. They may even have some memory cells left. All right. Whoops. Oh, sorry. So here what I want to show you are the cytokines. And I think you probably all have seen, you know, in the news that the cytokine storms are associated with worse symptoms. And that's what we were showing here, is that in red, the women who have symptoms have higher levels of cytokines, whether it's interferon gamma, IL-4, IL-6, or IL-12. So very simple, these are the women divided out by whether they have symptoms or whether they don't have symptoms. And each little dot represents a woman, all right? So you would say in general, yep, even in the milk, these cytokines are present. Not sure what they're going to do to the baby, but they're present. And women with symptoms have higher levels. What I want to say, though, in the next slide, oops, sorry, is when you look at this, and I'll ask you, what do you see? So if these represent all the women here and who had symptoms, and these are the women without symptoms, what do you see? I'm going to put my glasses on. Anybody want to comment? What is it? Higher cytokines, but what else within a group? Within a group, what do you see? I think I heard somebody say variability. Yeah. So again, you could be this woman here with symptoms, and actually your levels are at the level of somebody without symptoms, and here you have no symptoms, but you have pretty high cytokine levels. And so again, there's so much variability in the biology of what we see, and understanding it, interpreting it, what it means for your health, for the baby's health, is really still very challenging. There's a lot that we don't know. Okay, we're going to sum up that study, colostrum spot card study. We showed that the antibodies and the cytokines can be measured in colostrum collected on paper. The response was primarily IgA, sorry, it was a little stronger in IgA, right? The antibody response varies greatly among women, which may be why some babies get sick and some don't. And the levels of cytokines are associated with symptoms. Okay, if no questions there, we're going to go on. Yeah, great.
Come over there.
But you can shout or you can go over there.
So, seeing an increase in symptoms in babies, GI symptoms in babies whose moms who've had COVID since like 37 weeks and on. Is there anything that we can do to address the cytokines that are in moms to lower them so that babies are having less symptoms?
Oh gosh. Okay, so baby moms who had COVID 37 weeks gestation or later.
Or later, yes.
And you're seeing, and these are breastfed moms or?
These are breastfed babies that
breastfed.
have tummy problems, like really bad tummy problems that are not able to be resolved with like all of the normal things.
Okay, so I don't know the answer to reducing cytokines. That's a good question. I don't know the answer. You could measure them, but actually I would have thought that those moms in their milk would have antibodies that would really, they'd have sufficient antibodies, I'm going to show you, that I thought would have protected the entire digestive system. So, you know, maybe it's a combination of moms with low antibodies and higher cytokines. It's great to know, but a great question, but I don't have the answer. Was there another question? No?
No, I was going to take the mic to her.
Okay. Good. Maybe something I say later will speak to that, but I'll think about it some more. Okay, so yeah, in the second study is a longitudinal study, and we again looked at antibodies and neutralization assay and cytokines, and I'll talk about, and the memory cells, but I'll talk about 3 of these. Okay, this is a complicated study because we really wanted to look at the duration of the response, and we also did want to look at infant stool, which relates to your question. What antibodies are in infant stool, and are they protective? And we also wanted to compare this with the mother's blood. So this is maternal blood, not infant blood. We didn't want to prick the babies. And all of this was done by, I have a website for my breast cancer research, and so I just posted early on in COVID that I was doing this study, and I sent participants kits, just like the one I'll show you later, and they mailed this all back to me. So the women had to keep these milk samples that they collected in their freezer, and then they mailed them once here, and then they mailed them a second time. Okay, this is what I'm going to talk about now. It's a little funky, and I can't explain a lot about it, but we had decided to look at it. So I'm going to try to take you through this. So there's, again, all of the people who work in lactation probably know this much better than I do. It's called the breast permeability. And it's often measured by the level of sodium and potassium in the milk. And it also can be an indication of subclinical mastitis. Can you hear me correctly? Oh, okay. Funny. So what I want to point out in this is that there are two sets of labels over here. This is the scale for the antibodies, and this is a scale for the sodium ions over the potassium ions in the milk. And most women, it's thought after secretory activation occurs, so later on when they're breastfeeding, they have a very closed, so their epithelial barrier is nice and tight, and it's keeping out things from the blood. It's very selective now what gets into the milk. And so this low ratio here, it's actually, these are donations over 36 days, is nice and low in this woman's left breast. And her antibodies, you can see on this dotted line, she has a nice, a healthy IgA and IgG response on all of those days. IgM is a little bit right on the line, so it's not a positive. That's her left breast. Let's take a look, and so that's a very, her ratio there would be about 0.4 for sodium to potassium, a very healthy, nice permeability. Let's take a look at what happens to her. So keep in mind here that this gray bar here, this is the black here, this is her sodium potassium. Wow, it's gone wild. So maybe she has mastitis, but she didn't have mastitis. Not quite sure what's going on. She didn't complain, giving us milk, checked no mastitis, but you can also see that both the IgM now is above background, it's positive, so antibodies are getting in, whether antibodies are coming in from the blood or whether she's just producing more with this change in sodium potassium, I'm not positive, but I think they're coming in from the blood. And so is her IgG. And take a look at her IgA. So her IgA is just tracking perfectly with her permeability of her breast. And so this was one of the things that just clicked on us that, wow, when we want to measure things in the milk, because I do want to measure them for breast cancer risk, I probably should be thinking about the breast epithelium permeability. It just says we have a blood-brain barrier permeability. We have the breast epithelial. So, and this could explain differences among women. So I'm not quite sure, but there might be things one could check if in the milk, in terms of the permeability. That's something that's relatively easy to check, and that might be helpful for antibody levels or cytokines. And I should say also cytokines track too. So cytokines will increase significantly with increased permeability. Okay, so this is a neutralization assay, and so these would be your controls down here. And what we're really looking at is whether it was done a time course of these different times, and whether you see an increase. And so for both spike, what we call the wild type, the original virus, coronavirus, and for 4 of the variants, we can say that the levels of antibodies that I just showed you that are in the milk can neutralize. And this is called an inhibition assay, and so they are neutralizing. Great. And the last thing I want to show you is because I talked about memory T cells. And so we looked at memory T cells at day 35 and at day 120 in the milk. So you can see, I think there are about 8 women represented here, and somewhat more represented here. And so you can see, we said, wow, this is great, this is amazing. The memory T cells, those effector memory T cells that are going to stick around in the women are getting into the milk. Potentially, they could get into the baby through the gut, and they're sticking around maybe for 6 or 9 months, and this is really, really protective. And once again, I have just a take-home message here on the next slide. So all of these women, there are more here. We only had 4 who gave us milk at both times. So let's look at those 4, the only 4 who gave us milk. Okay, so what do you see here? Tell me something. Oh, yes, please. They went up. Well, here they went up, but here they went up, and we would have said everybody goes up, right? But what you see over here is it went up, and she went down. And so again, what I would emphasize here is variability. There's just a lot of variability among women over time, and that's something that we have to take into consideration. Okay, last study. In this last study, we wanted to look at the response to the vaccine. And it's another longitudinal study, similar to the other design that I just showed you, so I won't show you again. But we're only going to look at one thing here. I want to show you some different ways that we look at antibodies. And again, just to keep you interested, the reason why I want to show you these is so we can talk about the study we're doing here at Rose and how we might run better studies in the future. Okay, so let's look at some antibodies. So here what we're looking at is a little bit different. We're looking at the blood of vaccinated women, and we're going to say it's significantly increased for anti-RBD IgG, right? So not IgA. And here's the line that says you have to be above that line to be positive. And here's the pre-pandemic dried blood spot after the first dose and then after the second dose. And you can see all of the women, so that's comforting, right? Every mother in the study who was lactating had a strong response in her blood. So that was good. Now let's look in the milk. These are the vaccinated women. Here's their pre, and this is after the boost. We took the one where they have the strongest response, and this actually is not significantly increased. So IgA, very different from if a woman had COVID. IgA, for these 30 women, left and right breasts, after they're vaccinated, it's higher, and for some it's probably definitely a lot higher. But overall, the group is not higher, whereas IgG is higher. And that's really due to the vaccine. The vaccine is an intramuscular, right? And it's going to enter the cells, and it's going to enter the circulatory system and get into the milk. Whereas getting COVID is going in through the mucosal, and that is, and your mucosal organs produce more IgA. So it makes a lot of sense. And whoops, I meant to put this one up. And this is just to remind you, this is what we saw in the women who had COVID, that IgA was higher. Yeah.
How many days after?
That's after the second shot. This was a Moderna and the Pfizer.
Do you think that these numbers change with time, like as the vaccine becomes ineffective,
It definitely changes in time. I think we tried to pick the time that it was highest here, where we expected it to be highest. But you never know, there's variability, and these women here could just take longer for what's called class switching for their IgM to switch over. So absolutely. That was good. Yeah, please.
I know that it was recommended, especially for children, to space out the doses of the vaccine between the first and the second for a stronger immune response. Now, with these women, was it the typical, like, 2 to 3 weeks between doses?
It was. So it was early on, and these were, the women who got vaccinated were among the very first healthcare workers to get vaccinated. Again, they were across the nation, and so it was, yeah, I think that could be it. So for sure, you're bringing up good things. There papers are coming out, but we need to study the response in breast milk if we want to know what we have to do to increase that response. But you're right, yeah. Oh, I will try to do that. Yep, okay.
So I was curious, were any of the women who may have had COVID earlier on before vaccine were, do you see a change in greatness after baby was born?
Could you repeat that question?
So women who were pregnant earlier on with COVID and then gave birth, and a part of this, were they a part of this study, number one? And then if they were the changes significant from, or would it be the same result?
Yeah, so there's a great question. If a woman had COVID while she was pregnant early on, and then later got vaccinated, how would that affect the response that I was seeing? And were any of these the same women? So no, they weren't. I was conducting this study at the same time, 2 different groups. The woman who had COVID was before the vaccine was out. And then it is true that 2 of the women who had the vaccine had COVID. They do have stronger responses. So the combination of the COVID and vaccine seems to strengthen. So the one was off the charts in IgA and IgM. Yeah, that's good. No? Okay, great. Okay. Now I'm going to talk about, we collected stool, and what we wanted to look at is, are there antibodies in this stool? So you might ask, like, why are you looking at that? Well, we know when the antibodies go into, when the woman breastfeeds, the antibodies are going up and they're getting into the salivary glands, they're getting into the infant's mouth, so they really are protective. We know they're all the way down to the stomach, but how long can those antibodies survive? And so antibodies are pretty robust, and so we found that 1/3 of the infants had strong neutralizing antibodies still present in the time that they, in their stool. So, I think that's important. I think it's very protective. I don't know why the other 2/3 don't have those antibodies, and that might be related. It could be that the antibodies were degraded in some cases. It could be something I did not collect, which is a bit of an embarrassment. I did not ask if babies were being exclusively breastfed. We didn't see what I would have expected, that older babies had less antibodies. I thought, okay, maybe they're getting other food, but that wasn't the case. So it's a small sample size. But okay, great. So I'm going to give the summary of the studies in milk. And so most lactating women have been vaccinated, or most who have been have antibodies, and the response differs a little bit based on whether they've had COVID or they've been vaccinated. It's either IgA if you've gotten sick, a little bit more IgG. Varies greatly among women. And the antibodies do neutralize, but again, it's really significantly varies. We think the baby is definitely receiving protection from the milk, so I don't know anyone who's arguing not to breastfeed, even though there could be cases when the cytokines are high and are a bit of a problem. And more research is needed to understand the role of the cytokines in those memory T cells. So, great, I have some time left. So, I would take questions again on that before moving to this next and last part. Yes, please. Thank you. We're conducting, well, I spoke with Kim Marie. I was initially going to give a talk on something different, and speaking with Kim Marie, we said, wait, COVID is more important. Let's change that, and let's see if we can do a study. So I'm so excited to tell you right now this study that we started here. It's small, but I'll tell you about this, and yeah. So with Kimberly and Haley and Linda Sabecco, my colleague back at University of Massachusetts, we decided to do a study here. And why? Well, let's take a look for a moment. Of the major studies published, you can see, and even my own, I'm sorry to say, they have very few black mothers. So here are the sample size of the studies. These are primarily COVID studies. These are primarily vaccine studies. And here are the number of black or African American women included in the studies. They weren't excluded for any reason. I just posted my study online, and actually these are my 3 studies, and in this one, it's a woman who contacted me and she said, hey, I bet you don't have many black women. I said, no, I don't have any. She said, all right, I'll send you my milk. So given the variability that I just told you is out there, we should do the study. So we spoke, and again, the team out here, and I probably don't know all the names of the people who got involved, but we got approval at the University of Massachusetts, and within, just a wicked short time, Rose produced this and put this on Twitter, or their tweet, or wherever they put stuff. I'm not a social media. I know nothing. Okay. And then what we had is it would lead to this jet form, and the point is we talked about it, and we said, let's take samples from women if they had COVID, if they were vaccinated, or if they think they never had COVID and they were not vaccinated. Let's just go see. Okay. So this overall study design was to recruit black breastfeeding moms living in New Orleans. We'd send them the kits for milk, saliva, and blood. We'd ask them to deliver them here to the conference, which is a big ask, right? They live somewhere here, that's pretty. And then we have questionnaires, like we need all these questionnaires. And we got a little bit of money from my university when I presented it to them. They said, yeah, we can give you $4,000. So anyway, we can pay the women. And let me just tell you where we are to date, actually, yeah. So where we are, we had 23 women answer this. We had 12, this was as of last night, 12 signed the consent form, and we have 11 sample collections scheduled right here. Since then, since last night, 4 more women have said they want to give us milk. So it's really great, and if you add that 11 and 4, that's more black women than in all those other studies. Thank you. All those other studies I showed you combined. So, yeah, we're pretty excited. But you might say, like, why saliva and blood? All but 2 participants, I think, said they'd give saliva and blood. And for this, for the blood, it's sort of clear. The antibody levels between blood and milk differ, and so it's good, like, as a scientist, we're trying to figure out how that works. But particularly, we want to know if there is no response in milk, was there still a response in blood, and what does that mean? And then with the saliva, we want to know, well, the salivary glands and breast are more similar in being mucus, producing the mucus antibodies, and so we really wanted to see if that was there. And because saliva is some people don't want to give their blood, and as a second body fluid, even though it's just a little finger prick, the saliva would be a good fluid that we could collect from everyone, and we could collect it from men. Kim Marie was suggesting we do that study here. There is one more thing I want to say, and maybe this, I want to show you what we'll do in the lab. And some of the women have asked for results back, and we will give them the results. Any woman who wants her results back will give them, with the understanding that this is research, right? It's just basic research. So of course we'll measure the anti-SARS-CoV-2 RBD, IgA, IgG, and we'll do it in all the milk, saliva. But one of the things I didn't mention is, all the vaccines to date have been made against this protein. But if you have an antibody and you can measure, or you have a a protein, and you can measure any antibodies against this nucleocapsid protein, well, that's actually an indication that the woman had COVID. At some time, she doesn't know she had COVID, and that's what we think. So that would be specific, since this nucleocapsid protein was never used in any vaccine, it's specific to having COVID, not the vaccine. So we'll be measuring that also. Then the other thing we'll do is we'll do neutralization assays. So we'll be the first time then with this study to do both types of inhibition assays. We will measure the cytokines in milk just because we want to know, and we can do it from that, we can do it in these samples. And we'll measure breast permeability. 3 minutes, okay. Well, then I'll go back to this. So what I want to show you is we're still collecting, so we're up to 15 today, but we're until, thank you, until tomorrow night, we can collect more. And it's just this little kit. If anybody wants to, we give them this little bag here and little, to prick their fingers and put their blood on this card. And these little, they fill this with milk. Left, right breast, and just the saliva to there. Up to that little line. So we're hoping that we might get more and go home with a few more and be able to give, finish. Oh, thank you, Linda. Yeah, you definitely do. In fact, if you gave a fraction of it, it would still be fine, because remember, I could truly do it from a piece of paper.